Hello, my name is Scott Requadt, CEO of Talaris Therapeutics. I'm very pleased to present Talaris today at Meeting on the Mesa. This is our standard forward-looking statement disclaimer, which I'm sure you can all read very carefully. I'm going to start with an audacious statement, which is that we believe with our therapy and a single basic underlying biology, we can potentially impact three large areas of unmet need. Firstly, to reprogram the immune system of a transplant recipient to see a transplanted organ as self. Secondly, to potentially restore tolerance to self in patients with a severe autoimmune disease. And thirdly, potentially to serve as an alternative to traditional allogeneic stem cell transplant in replacing diseased blood or immune cells without the need for full myeloablative conditioning. Our lead program, FCR001, is currently in an open-label phase 3 study, the goal of which is to induce durable immune tolerance to a donated kidney in a living donor kidney transplant recipient. I'll be sharing with you compelling phase 2 data that we've generated over the course of the last 10 years. And over the course of that study, we've identified what we believe to be a highly predictive surrogate marker, both to predict the ability to come off immunosuppression and the likelihood of staying off immunosuppression. I'll share with you some of the phase 3 results from that study. I'll also talk briefly about a couple of additional indications, which we've now begun phase 2 studies in order to assess the extensibility of our approach to other areas of unmet need. We do all of our cell therapy processing and analytics at our own in-house cGMP processing facility. And I'd like to mention that we have, as at the end of June, over $207 million of cash on hand, cash taking us through the end of 2024, fully funding all of our programs. This is a quick snapshot of our experienced leadership team and board. Nancy Krieger, our Chief Medical Officer, is a transplant surgeon by training and has been involved in this program since its prior collaborations with Novartis. And Mike Zanowski, our Chief Technology Officer, has over 20 years of experience in CMC, the last 13 of which have been entirely in allogeneic cell therapies. And now I'll go on to the key highlights from our phase 2 study, which were the basis of the RMAT designation that we received from the FDA and our now ongoing phase 3 study. We've conducted a phase 2 study in 37 living donor kidney transplant recipients, and in those recipients, we were able to get 70% of them durably off all of their immunosuppression therapies, on average about 1 year after their kidney transplant. We've been able to do this across all degrees of immune system mismatches between donor and recipient. And once we optimized the trial protocol, we were able to achieve tolerance in 14 of the last 17 patients. And it's this protocol that we've now taken forward into the phase 3. Notably, every single patient that we were able to get off immunosuppression has stayed off immunosuppression durably for the duration of follow-up, with the median follow-up now being over 7 years, and the longest of these patients now being at 12 years from their transplant. Also notably, 10 of these patients had a pre-existing kidney-related autoimmune disease, such as FSGS or IgA nephropathy, and we were able to tolerize 7 of those 10 patients to their donated kidney. And notwithstanding the fact that recurrence of these autoimmune conditions is frequently noted in these patients, none of the 7 patients that we tolerized to their kidney have experienced recurrence of their kidney-related autoimmune disease. And we think that this is an interesting initial proof of concept of the extensibility of our approach into the autoimmune space. The biology that we're leveraging is a biology that dates back to the 50s and in fact was the basis of a Nobel Prize for Peter Medawar in 1960, and is premised upon the concept of enabling hematopoietic stem cells from an unrelated donor to be placed into the bone marrow of a recipient under conditions that allow the stem cells of both the recipient and the donor to coexist and essentially mature into mutually tolerated functional immune and blood cells that see one another as self, creating a state of what we refer to as allogeneic tolerance. But instead of doing a traditional allogeneic stem cell transplant, our product focuses on a subset of cells that we believe need to be present in particular concentrations in order to enable engraftment of the donor stem cells in the recipient and to do so with a non-myeloablative conditioning. And we call that product FCR001. And the donor of the cells is the same donor as the kidney donor. FCR001 itself comprises 3 critical cell components that we control for in our product. The first of these is a hematopoietic stem cell, which of course is the progenitor cell for both blood and immune cells. The second of these is a unique cell that was discovered by our founder, Dr. Suzanne Iltstadt, called a facilitating cell, which we believe has unique properties both in terms of promoting engraftment of stem cells in an unrelated recipient and also upregulating various tolerogenic pathways, enabling allogeneic tolerance as these cells engraft. And finally, we have a small amount of alpha beta T cells from the donor, which are also included in the product, which we believe are critical to engraftment and counteracting host versus graft response, since we're not fully ablating the immune system of the recipient at the time that we do this transplant. We work the product in our own in-house facility, and the entire product involving all processing can be completed soup to nuts in less than a day. We don't do any viral vectors or genetic engineering, nor any cell expansion steps. And all of this work is done in-house at our own cGMP facility. And importantly, no manufacturing processes or controls that were critical were changed between phase 2 and phase 3. This is a quick snapshot of our pipeline, the lead program of which, as I mentioned, is focused on living donor kidney transplant. We also have 2 other phase 2s underway, which I'll describe shortly, and the potential to extend this across other areas such as deceased donor organ transplantation, as well as other severe immune or blood or metabolic disorders. Our lead indication, as I mentioned, focuses on reprogramming the immune system of the transplant recipient to see the organ as self without the need for immunosuppression. You're probably very familiar with the fact that if you receive a transplant, you normally need to be on a cocktail of immunosuppressive medications for the rest of your life or the life of your transplant, or you will reject the transplant. And unfortunately, these drugs themselves are toxic to the kidney, and as a result, contribute to kidney function decline and the need for retransplant. Typically, most transplants lasting only 10 to 15 years. Because these subjects are chronically immunosuppressed, they're also at much higher risks of cancer. There's a host of cardiovascular comorbidities associated with chronic use of these drugs, risks of infection, significant quality of life burden. And our goal is to free these individuals of the need for this cocktail of immunosuppressive agents and to live a normal life with an intact immune system, not rejecting their organ. I won't recapitulate these data other than to remind you that this is what we were able to accomplish in phase 2. And in this slide, we show what the degree of HLA or immune system matching was from the donor to the recipient. And what you see is that the vast majority of subjects in our phase 2 that we were able to tolerize to their donated organ by getting engraftment of the donor stem cells in the recipient, such that the immune system of the recipient recognized the organ as host, were actually quite profoundly HLA mismatched between the donor and the recipient, many of them being 2 or even less out of 6 matches. What we've also observed from our phase 2 is, as you would expect, if you were to remove the immunosuppressive agents, which are nephrotoxic, you would expect to see well-preserved kidney function over time. And that's what you can see from the line graph on the left. The blue line being intent to treat, and the orange line being the kidney function over time of those patients that we were able to wean fully off their immunosuppression. And what we've observed in our phase 2 is very well-preserved kidney function in these patients, whereas by contrast, the gray line shows the kidney function decline over time in a retrospective analysis of a cohort of patients who were concurrently transplanted at the same center in the same time frame and would have met our entry criteria. On the right side, you see the rate of patients who were tolerized with our therapy to their donated organ, and what % of those are on antihypertensives or dyslipidemic agents, which compares very favorably to the bars that you see in orange, which is what's typically observed in standard of care and was observed in that retrospective cohort analysis. I mentioned earlier that we've identified over the course of the phase 2 a surrogate marker that we believe is highly predictive of the ability to wean off immunosuppression and stay off immunosuppression. If you think back to the biology of what we're trying to do, we're trying to get the donor stem cells to engraft in the recipient and create a donor-derived immune system that sees the donor's organ as the recipient, the donor's organ in the recipient as self. And we can measure whether or not we've been successful in getting engraftment of the donor stem cells simply by sampling blood from the recipient at various time points post-transplant and looking at what percent of the T cells in that blood actually have the donor's DNA. And if 70% of those T cells have the donor's DNA, we would refer to that individual as being 70% donor T cell chimeric. In our phase 2 and in our phase 3, if a patient is able to achieve better than 50% donor T cell chimerism by month 6 after their transplant, this is what triggers the beginning of removal of immunosuppression from the recipient over the course of the next 6 months until, if all goes well, they'd be totally weaned off all immunosuppression at month 12. And what we've observed here, and this is the data from our phase 2, is that if we're able to achieve chimerism in the recipients, typically we've achieved extremely high levels of chimerism. And once established, that chimerism was highly durable all the way up to 10 years. Not a single patient who had chimerism at 12 months has lost chimerism for the duration of follow-up. And that's been synonymous with our ability to maintain tolerance to the donated organ in these patients. And even if patients had chimerism as early as month 3 post-transplant, 90% of those patients were ultimately able to be weaned off immunosuppression, and all of those stayed off. So we believe chimerism is a highly predictive surrogate, which we think has the potential to enable us to predict which patients are going to be able to wean. That sets the stage for the phase 3 registration study, which is now underway. It's an open label study in which we're enrolling a total of 120 donor recipient pairs, 80 of which will receive our FCR treatment and 40 of which will receive a standard of care kidney transplant. The protocol, as I mentioned, is almost identical to the phase 2 protocol that we optimized. And our primary endpoint is what proportion of the 80 patients who receive our therapy are off all chronic immunosuppression without a rejection event at 2 years post their transplant, which is typically 1 year after they've been weaned. Our secondary endpoint is confirming that there's no meaningful decline in kidney function associated with our conditioning or with our therapy. In neither the primary nor in the secondary endpoint are we comparing this to the standard of care arm. Rather, we're just looking to make sure that the lower end of the confidence interval in the case of the primary endpoint is above 30% off immunosuppression, and in the case of the second endpoint is above a 5-point decline in eGFR. And based on our phase 2 data, we believe we're very well powered to see significance on both of those endpoints if the product performs as expected. We currently have 17 active sites in the United States, spanning the coasts as well as the Midwest, that are actively enrolling. And this is the phase 3 update that we recently presented shortly after ATC. As at June 15th, which was the data cutoff date, we had enrolled a total of 22 donor recipient pairs in the study across 10 different clinical sites. And 5 of those sites have actually dosed a total of 7 different donor recipient pairs. Now, given the 2-to-1 randomization, 13 of those pairs have been randomized to active, 8 to control. And of those 13, 7 had been dosed and 1 was pending at the time of the update. The mismatched data of the donor recipient pairs continues to be significantly mismatched, as was the case in the phase 2, and that's shown in the histogram on the right. We were pleased to observe that of the 7 patients that we've dosed to date, all 7 of those have engrafted the donor's hematopoietic stem cells. And of the 6 that have now been followed out to at least one time point at which chimerism is being measured and reported, all 6 of these have better than 50% T cell chimerism at every measured time point. In fact, 3 of these patients are now more than 12 months out from their initial transplant. And per protocol, all 3 of these patients have been fully weaned off all of their anti-rejection drugs per protocol. We have 2 more patients who are beyond their 6-month visit and 1 more beyond the 3-month visit, all once again better than 50% T cell chimeric, which in phase 2 was highly predictive of the ability to come off immunosuppression. None of these patients have experienced any rejection events, nor even any donor-specific antibody, and all have maintained stable kidney function for the duration of follow-up. From a safety perspective, the adverse and serious adverse events observed have been consistent with what you generally expect in a kidney and allogeneic stem cell transplantation involving non-myeloablative conditioning across HLA mismatch barriers. We did observe 3 cases of low-grade acute graft versus host disease, all of which were fully responsive to treatment and in fact fully resolved as at the data cutoff date. And in fact, one of those cases, which was more than 12 months out post-transplant, was able to discontinue all of their anti-rejection therapies, notwithstanding the fully resolved acute graft versus host disease. We have one of those 3 cases that did go on to develop a moderate chronic graft versus host disease, which has also been treatment responsive. And the study continues to enroll, and no stopping rules were triggered. Notwithstanding all of these things, because the incidence of graft versus host disease in these first patients was higher than what we observed in phase 2, we did review if there were any significant changes between phase 2 and phase 3 that might have been associated with this. And we did identify a change in an increased use of a mobilization agent called plerixafor. As a result, we have modified our trial protocol to eliminate plerixafor as a mobilization agent in all of our donors going forward. That frankly does align us better with the standard of care collection practices. And we've also added one more post-transplant dose of Cytoxan, which has emerged in the past years as a gold standard for the reduction of risk of graft versus host disease. And we're optimistic that these steps will result in a lower rate of graft versus host disease going forward. Notwithstanding that all of these were treatment responsive and low grade, we'd like to see that even lower. Beyond that, we are now investigating the potential of this therapy to be extended into both patients who've previously received a living donor kidney transplant up to 12 months prior to see if we can use the same product made in the same way with cells from the same donor, but inducing tolerance after the transplant. And this is a stepping stone to potentially opening up the potential in the deceased donor space, where we would be looking to obtain cells from the deceased donor concurrently with the organ being transplanted into the deceased donor recipient. And then a few months after that, infusing a product similar to FCR001 and comprising the same types of cells taken directly from the bone marrow of the deceased donor as a means of inducing tolerance to the deceased donor organ. And that, of course, is a much larger opportunity in terms of patients and a tremendous need. Finally, the other phase 2 that we've recently initiated is our Freedom-3 study, and this is targeting individuals with a form of scleroderma, which is rapidly progressive and for which there is no disease-modifying or curative therapy. And in particular, we like this indication because autologous stem cell transplant has already been shown to induce a durable remission in a fairly high % of these patients. But the caveat is that these patients require their own cells to be given back to them, which we already know have a proclivity to making immune cells that can be fooled into attacking the self, but also the whole therapy must be administered using full ablative conditioning, which can't be tolerated very well by many patients. We believe there's an opportunity to leverage and extend this biology with our therapy, which would involve using healthy donor stem cells and involves the use of a non-myeloablative conditioning, which profoundly would improve the potential risk-benefit profile for these patients. We expect to provide our first report on this in 2023. In summary, using the same biology and the same product, we believe that we can address multiple areas of very high unmet need. In particular, starting with living donor kidney transplant, in which we're looking at both concurrent as well as up to 12 months delayed administration of our product with the kidney transplant as a means of addressing a very significant initial opportunity in kidneys, which are, of course, the most frequently transplanted organ in the United States and worldwide. But we think the same biology can be used to restore tolerance to self in severe autoimmune disease, with scleroderma being our initial indication, and can potentially be used extensively in other areas where allogeneic stem cell transplant has been used to replace diseased or modified cells, but where limitations such as HLA matching, as well as the need for full ablative conditioning, have lessened the use of that, both of which we could potentially obviate. We see a number of opportunities to extend this biology into other areas of unmet need and create value across all of these areas. So in summary, Talaris is developing a single-dose investigational cell therapy, which we think can transform the standard of care in a number of areas. Our lead program now in a registration phase 3 with very encouraging initial clinical results that we'll continue to report on next year, and in now phase 2 studies in 2 indications, which we believe will begin to show the extensibility of this approach in other areas of unmet need. We do all of the manufacturing in-house in our own facility, and we're very well positioned from a cash perspective to drive through multiple clinical readouts over the next several years. Thank you so much for your attention and for your interest in Talaris.